I'm delighted that Dr. Sam Gandy has agreed to present today as part of the Cyber series. Sam really needs no introduction, nonetheless. He's the Mount Sinai Professor for Alzheimer's disease research, and Sam really is a worldwide authority, a very well-known authority on Alzheimer's disease and the pathophysiology of Alzheimer's, and has made really seminal contributions. Sam is also an authority on potential therapeutics. So I'm very keen, we're just discussing this, very keen to get his perspective on the very recently FDA approved new therapeutic for Alzheimer's disease, aducanumab, I believe is how it's pronounced. So the timing of Sam's presentation here today is spectacular. And so without further ado, I guess I'll pass over to you, Sam, so we can hear about your work.
All right, Paul. Thanks very much for that kind introduction. Yes, this is an unusual week to be lecturing about the pathogenesis of Alzheimer's disease with the FDA approval of aducanumab on just this past Monday. And let's see if I can get this to advance. Here we go. So this reminds me of 2015. Mary Sano and I did this. You see the masthead here for paper for an opinion paper review for solanezumab when it was being contemplated, but unlike sola, which was not approved, aducanumab, also known now as Aduhelm, under the brand name, has been approved and is immediately shipping, going to be available in 2 weeks for a cash price of $56,000 out of pocket so far, not really evaluated for third-party reimbursement. So this is like a solanezumab deja vu, but worse, because unfortunately, as was true with sola, as you see from the headline, and as you'll hear in the subsequent slides, there's no obvious meaningful benefit for patients that might lead them or their families to consistently see any improvement in their function. And in fact, the companies aren't claiming improvement. The issue is whether this might slow progression over the coming years. So the current batch sort of generation of anti-amyloid antibodies have been selected or designed to be specific for the amyloid beta oligomers. And I'll get into detail on what those are in just a moment. So aducanumab, now patented under the name Aduhelm from Biogen, is co-marketed with Eisai. Donanemab is currently actively under trials from Eli Lilly. And you may have heard of an antibody called BAN2401 or lecanemab, which is co-marketed by BioArctic and Eisai. These are all, not only are they specific for oligomers, but they are designed to be so. They have biomarker benefits that are far superior to the prior generations. And that is that these antibodies all convert amyloid scans, a patient's amyloid scan, from positive to negative fairly rapidly over a period of weeks to months. But there's a huge problem still that remains, and despite this biomarker benefit, they do not show improved cognition. And even the patients who show statistically significant slowing of cognitive benefit are identified by formal testing. And so in general, neither the patients nor their families consistently observe any objective evidence of benefit. Now, there are still ways that this can work out well. For example, over 5 to 10 years of therapy, perhaps these function curves will diverge and we'll see a meaningful benefit. But that's not usually the way drugs are approved. So why do we continue to, there's the amyloid hypothesis has been very divisive and controversial, and this is a figure that Michelle Ehrlich and I used in a review or an insight in the Journal of Experimental Medicine that appeared just 2 weeks ago. And now there are 27 mutations in the amyloid precursor gene alone in and around the A beta domain that are all linked to familial Alzheimer's disease. So it simply is not tenable to say this is not a way that Alzheimer's happens. At least sometimes, albeit rarely, it begins with amyloid accumulation. And in general, the most toxic species appears to be these oligomers. So unlike the fibrils that you're used to seeing in terms of being congophilic and basically the defining biophysical form of amyloid, these are amorphous clumps that are the most potent toxins. Now, we've been working on oligomers for a while now. This is the masthead from a paper in Annals of Neurology in 2010, a mouse that Michelle created from the very first amyloid-causing mutation that was discovered by Efrat Levy and Blas Frangioni at NYU as the causative mutation underlying hereditary cerebral hemorrhage with amyloidosis of the Dutch type, or Dutch CAA, cerebral amyloid angiopathy. It's covered. The paper was somewhat surprising or shocking in that these are mice that accumulate amyloid beta oligomers, that is with the Dutch mutation, and never ever get fibrillar plaques for their entire lives, up to 2 years of life. AARP covered it very nicely on its cover of its bulletin, then the surprise that could change everything. So it changed some things, but it hasn't really changed enough yet. And part of that's because these oligomers are so challenging to work with, and you'll see why that's the case. So histologically, these mice accumulate material that seems to be in electron-dense late compartments of the central vacuolar pathway. So again, no detectable plaque pathology ever, no tinctorial amyloidosis ever, no tauopathy. So what you see here is the neoepitope generated by the generation of the A beta 42 C terminus that's immunolocalized to these multivesicular bodies that are endosomal lysosomal components. So again, we know that we describe these as the Dutch A beta oligomer only mice because they don't have plaques, they don't have tangles. The figures that we see in these mice are similar to those reported by others. The figure at the top is from a mouse created by Matthias Schuker and Takeyomi Saito that expresses the Arctic mutation. Arctic mutation of APP is at the same sequence, at the same position as the Dutch 693, but the Dutch is E to Q and the Arctic is E to G. But here you see the accumulation of immunoreactivity in perinuclear vesicles that look like TGN and endosomes and lysosomes. The Dutch A beta oligomer only mouse that Michelle created is there at the bottom. Use my little pointer here. So these are our mice stained with 6E10 and 4G8 standard antibodies, and these patterns look very similar to what Matthias and Takeyomi reported. If you compare them with, say, the lower panels here from the Shao mice with Swedish APP, they also show perinuclear vesicular immunoreactivity. I think that ours are the expression level of the protein in Michelle's mice is greater than the Shao mice. We have done some experiments where we cross these Dutch oligomer-only mice with mice containing the mutant presenilin molecule. That then converted them to plaque formers. So now they have congophilic amyloid, but their learning defect was no better or no worse. So converting the biophysical form was not clinically important to the mouse. The most important thing seemed to be the presence of the oligomers. We can't exclude the possibility that there might be some contribution from the vasculature. They do have pearl stain positivity in the microglia around blood vessels. So there can be some leakage, and their vessels do stain with A beta, anti-A beta antibodies. So they do accumulate some material there, just not enough to get heavy congophilic angiopathy. Just to sort of go straight to the primary data to show what we observed and how we sort of worked through this. These are learning curves from water maze tests of either non-transgenics above or the Dutch oligomer only mice below. You can see over time these mice learn fairly well, and they're pretty tight, aside from this one yellow mouse. But the oligomer only mice here, their learning is all over the place. The noise is increased dramatically. Now, what we were able to do was to collaborate with, this was in the early oligomer days, collaborate with some colleagues at Merck who had developed an oligomer, or basically a dimer and above ELISA, where 6E10, an epitope there's only present in one copy in A beta, was used for both capture and detection assay, so that a 6E10 capture, 6E10 detection assay detects everything with a dimer and above. And when we looked at that, analyzed the extracts, there was a stratification of the what we saw in the non-transgenics, what we saw in mice that were transgene positive, but really without detectable oligomers, but here in mice with more robustly detectable oligomers. And you can see that that there is an intermediate phase where we could detect gene expression, but not yet detect oligomers with that ELISA. But yet, once the oligomers were detectable, that clearly showed that the mouse was more impaired. And the dose response you can see here of the three groups, 5 versus 7 versus 11. And those, if we then replotted those primary data, you could see that the learning behavior was roughly proportional to these three bins of oligomer level. Later, Elise Knight used the same Dutch oligomer-only mice to assess the ability of affinity purified gamma guard, that's human IVIG, to improve the function of these Dutch oligomer-only mice. And you can see, so this is a human IgG product that's sold and marketed for immunodeficiency. There were trials at some point for gamma guard, which failed clinically for Alzheimer's disease, but a fair amount of effort went into characterizing gamma guard, and we were able to show that they did in fact contain a fraction. If you took A beta oligomers and prepared an affinity column and then passed over that human gamma guard, IVIG, and eluted out the anti-oligomer antibodies, those would improve the novel object recognition function in these oligomer-only mice. Was not true for fibril affinity or for total material, only for the affinity purified, really concentrated product. But there is in gamma guard the product. There is a subpopulation of IgG that reacts with A beta oligomers. So to try and understand why these mice are impaired in a greater detail, Ben Reedhead, who's now an assistant professor at Arizona State, Jean-Vianney Aurier-Mirand, who's an instructor in the laboratory at Mount Sinai, together with Michelle, and in collaboration with Joel Dudley and Eric Schatt, performed transcriptomic analyses of our standard Dutch oligomer only mouse and compared that with a standard plaque forming mouse, the Borchelt double transgenic that expresses Swedish APP as with its mutated, and it's just a tandem missense mutation at lysine and methionine 670 and 671 are changed to asparagine and leucine. And these are double transgenics where the construct is created with this double transgene that migrates together during husbandry. And the presenilin 1 gene as well, that's deleted has exon 9 deleted, and is a pathogenic mutation for Alzheimer's disease. And you can see here that using bulk transgenics, bulk RNA sequencing, was possible to see a substantial signal from the oligomeric line that looked like it was promising. More recently, Emily Castrano in the laboratory has done single cell sequencing, single cell RNA-seq on these mice, and that has been less informative in terms of pointing us to inflammation, which is what we expected to find. And if you compare some of the DEGs from the two lines, the more of the DEGs were associated with the plaque-forming mouse, the standard Borcheldt mouse, than with the Dutch oligomer-only mouse. So again, this is consistent with the notion that in standard plaque-forming mice, the progression may well be due, or the symptoms may well be due to inflammation, but this is not clearly the case in the oligomer mouse. So some of the trickier parts of working with oligomers and of trying to understand the pathogenesis of Alzheimer's disease are associated or shown on the next couple of slides. So these are our PETs. So the top row are MRIs, the middle row are amyloid scans, and the bottom row there is our FDG, fluorodeoxyglucose. So there's a healthy control here. The far right, you see 3 subjects that have Alzheimer's disease, and that's have either mutations in the Swedish. You have the Swedish mutations in APP or in presenilin, or have sporadic disease, and they have what you see here as the signature accumulation of amyloid on their amyloid PET scan, while the normal control here, healthy control, has no amyloid accumulation. However, these 7 in the middle are all carriers of the Arctic mutation. They're all accumulating A beta oligomers. There are many impaired. These are carriers who are not yet impaired. And these are the non-carriers here in the middle. But both of these carriers, you don't see any difference, any appearance of their amyloid, any detection of the oligomeric amyloid in these subjects. Sorry, I misspoke on this. These are the non-carrier family members, and these are the carriers. So they do not show evidence of amyloid, despite the fact that their brains are rich with amyloid oligomers. It's just that they're oligomers of the Arctic type, so they don't bind the amyloid ligand. Now, to show you how this biophysical toxicity situation, how this dissociation occurs, these are peptides, these are synthetic amyloid peptides, either wild type or mutant, prepared in various mole fractions and then studied for toxicity here that you don't see on the left, in mole fractions of 25, 50, 75, and 100. And here, these very same preparations that have been studied for toxicity are examined under electron microscopy. As you can see, there's a wide variation in the morphology. These are oligomers. These scattered spheres are the toxic oligomers, while the more conventional amyloid fibrils are what you see here. So as you see here on the toxicity plot, the oligomers are the most toxic, and the fibrils are relatively non-toxic. However, if you look at amyloid scans, the oligomers are amyloid PET negative, while these are all PET positive. So this is part of the confound. If this is the poisonous species, the most poisonous species, and this is the one that's the least detectable by amyloid imaging, this is quite a challenge for us. And I think we're still hamstrung clinically with humans in the inability to sort this out in the living human brain. Moreover, there are even examples like this, where there's a linked molecule, in this case CR1, that's associated with the risky allele is associated with reduced amyloidosis, not enhanced. And if you compare here, this is just looking at the relative prevalence of amyloidosis in the low carriers. In the reference group versus the high risk group. And in every case, in every region of the brain, the high risk genotype is associated with almost invariably here as well in the posterior cingulate and anterior cingulate. The amyloid, the high risk allele is associated with a reduced amyloid burden. The formulation that we proposed, and this is a commentary with Harry Haratunian, Steve Dukoski, Mary Sano, and Eric Schatt, that appeared in 2013 in Biological Psychiatry, together with a paper from Madhav Thambasetti, who was reporting this surprising finding, and we gave our editorial here, the commentary, sort of tongue-in-cheek title of "Vanishing Amyloid Hypothesis," because in fact that seems to be what's going on. Our proposal in the commentary is that this is a clinical Alzheimer phenotype, looks indistinguishable from what we usually think of as being garden variety, sporadic, late onset sporadic Alzheimer's. But instead of the main driver of the clinical syndrome in these patients being the amyloid toxicity, it seems to be the inflammation. We would propose then that what's happening here is that inflammation has sort of overtaken, maybe is helping to clear away the amyloidosis, but is instead or is at the same time causing sort of a friendly fire bystander effect on neurons. Now, I sort of envisioned Alzheimer's for many years as having amyloidosis early, inflammation in the middle, and then tauopathy coming later. What's emerged recently from Hui Zhang and Daniel Geschwind is there is yet another group of inflammatory events that are triggered by the tauopathy in itself. They're not sort of triggered by amyloidosis and then on their way to tauopathy. These are disturbances of the inflammatory process and the interferon signaling system in particular that drive that seem to drive neurodegeneration from tauopathy. So this is we didn't really I didn't expect this from what I thought for many years, but it's clear this is also part of what makes the disease so challenging. Now, there's one more new wrinkle that's emerged that is also not obvious. What turns out to be the case, this has just been revealed in the last few months, is that TREM2, that we'll talk about in great detail in just a moment, is a microglial molecule that seems to play a role in restraining the accumulation of tauopathy downstream of amyloidosis. And we certainly knew that amyloidosis could invoke or could induce tauopathy, but I think no one would have thought that necessarily there was a microglial molecule, a specific microglial molecule that was playing an important role or playing a key role in determining how neurons develop tauopathy in response to amyloidosis. So I've sort of, in trying to get my arms around the disease, I've sort of invoked the model that Donald Rumsfeld used to talk about Iraq and Afghanistan, and that's the known unknowns and then the unknown unknowns. So we have these multiple variables, these sort of major event types that can have positive, negative, or neutral events. And we understand them poorly or not at all. And we don't know their temporal sequence or how to personalize in terms of individual patients. So the biophysical status and bioactivity of A beta is thing one. You have oligomers and fibrils. Some are toxic, some are not, some are maybe even protective. There are inflammatory processes that are invoked by A beta that may be good, bad, or indifferent. And now we know that there is inflammation induced by tauopathy that may be good, bad, or indifferent. The most recent evidence with TREM2 suggests that there are even some things we didn't expect coming. There's sort of the unknown unknowns, the A beta induction of tauopathy involving these unexpected pathways like TREM2. So there are a couple of other challenging issues as the clinical decline progresses, amyloidosis can increase, decrease, or remain unchanged. Inflammation can reduce amyloidosis, or it can cause, it can contribute to the decline. Moreover, it's very clear that once initiated, some of these events may be self-sustaining. That is, a beta can trigger inflammation, and that inflammation may be self-sustaining even after removal of amyloidosis. And I think that's the challenge that we're trying to deal with these immunotherapies for A beta. That is, it appears that there's certainly, maybe if we get in very early, they'll be more useful, but certainly at the point that we're getting in now, even getting them away doesn't prevent progression, but the progression and decline once initiated goes on. So one of the things that all the sort of distraction by or the issues about aducanumab take away from are reports like this one, which appeared in Neurology last month from Sweden. This large study of cholinesterase inhibitors with a very surprising result that you'll see here. That so this is looking at separate cholinesterase inhibitors and the progression to severe dementia: donepezil, rivastigmine, and galantamine. So there's no significant effect on progression to severe dementia by donepezil and rivastigmine, but galantamine has a substantial effect, and that's seems to be present in looking at death as well. The cholinesterase inhibitors all seem to reduce death and seem to reduce the appearance. Sorry, my things in the way. There's the p-value. So almost no one in this large study who was on galantamine progressed all the way to severe dementia. They seem to stop in the moderate stage. One would suggest that this is evidence for slowing for meaningful benefit on progression, but almost no one has been talking about this paper, and it's sort of a shame as we think of this compound that's infused and is unreimbursed and is expensive. On the one hand, while here's a safe, orally active, inexpensive compound that now studied 20 years after approval, looks more promising than we might have expected. So I think, so we can move on now to, so I've asked Mary Sano to be in the audience to see if she has any comments about aducanumab before we segue on into the microglial portion. Mary, are you there?
Sure. You know, I think the major challenge from the study that we saw, the report of an approval for an agent on a biomarker, is the fact that we don't have great connectivity to the symptomatology and the biomarker. And the word toxic here is also a very interesting one. I mean, it's a great model, but how do we hook that back to the clinical phenomenon? Because that's really what I feel that challenge is really the one that we haven't met. And we have tried to look, I know several in the audience look at different animal models of cognition and behavior, but in some cases they have not been tied to the amyloid pathology. So I guess that's, for me, a really big question about, as we look at these other mechanisms, how do we make sure that it's better hooked to the clinical condition? And I think that's really a big challenge here. So.
Okay, thanks. So in an effort to try and understand what's going on a bit better, I'm trying to check to make sure my camera is okay. My vision is off there. All right. So I want to introduce now work that's been going on in the lab for the last few years, largely led by Michelle Ehrlich together with Jean-Vianney Auré-Mirand on the AMP-AD effort established at Mount Sinai in collaboration under the leadership of Eric Schatt and Ben Zhang, who do the genomic discovery, and our unit works on the the validation. So let me tell you about AMP-AD. So this began in 2013 as a consortium specified and designed to be so by the NIA of big data groups where there were centers where there were in general ADRCs, Alzheimer's centers, brain banks with hundreds or thousands of accumulated specimens over decades. The ADRCs have been in existence for about 40 years now. So ours was one of the founding centers. It's a collaborator. There were collaborators between Brigham and Rush, Mayo and University of Florida, Harvard and Emory, UCLA group, and the Duke group. So in each case, these were units, centers, where there were brain banks and accumulated material by standard ADRC criteria, characterized and collected and harvested body fluids, cells, DNA. And in every case as well, there were big data groups that were able to perform computational analysis of multi-omic data really coming out of this these brain bank material. So together with the commissioning of the first cohort in what's called now AMP-AD1, there was also recruited the Sage Bionetworks group from Boston, who, while each of these regional centers was doing its own sort of big data thing with its ADRC, with plowing through Alzheimer brains, Sage Bionetworks was harmonizing and sort of organizing a database wherein all of the local big data groups or big data could be stored and probed as one large unit. That is, there were the individual, as though they were, the individuals could then be probed as though they were one large center. The platform was harmonized and made available generally to qualified investigators at no cost. So there are a number of these units now online. Synapse.org, Agora. There's now a group called the AD Knowledge Portal, which provides these updates and sort of as an umbrella for these various resources. And as well, at the same time, joining these AMP-AD groups through a public-private partnership through the foundation of the NIH, were Takeda, Lilly, Merck, GSK, and Biogen, among others. And so the overall task was to plow through all these brains at the sort of multi-omic level, analyze all the data computationally, and try to make some progress in understanding late onset sporadic Alzheimer's disease. So these are not the early onset autosomal dominant forms. This is now the garden variety forms of sporadic AD. And the idea with having the pharma partners involved was hundreds of hits were coming out of these computational analyses. They helped stratify or prioritize which hits might be druggable and which might be most accessible and which might be sort of too difficult. So this gives you just sort of a snapshot of the workflow in AMP-AD. So the total cohort that you see is summarized here. So the nearly 3,000 samples from almost 900 controls, 1,300 Alzheimer's, 168 MCIs, and then 500 and change with some other diagnoses. And these are, again, accumulated at multiple centers, analyzed by whole genome sequencing, by RNA sequencing, and by mass spec. And all those data are then crunched by the computational groups to look for networks. And this is the founding network for the Mount Sinai AMP-AD program. This was from Ben Zhang, who now leads the program, and it was developed by under the leadership of Eric Schadt, and together with Jun Zhu as well. So this is the complement subnetwork that you see here is part of 6 networks that emerged from the analysis that Ben Zhang and Eric Schadt reported in Cell in 2013, and the masthead of that paper is shown here below. This particular sub-network attracted the most attention because it had a number of inflammatory molecules and already known Alzheimer's associated molecules around. ITGB2, ITGAX, the complement receptor components, CD33, known as a GWAS gene, already was in the subnetwork. These complement components, C1q, A, B, and C, and C3, and C3AR1, which is both a complement pathway component and a receptor for VGF. At the center of this subnetwork, complement subnetwork, is a molecule TYROBP, stands for tyrosine kinase binding protein, also known as DAP12. So the TYROBP network is what we've been focusing on, and what I'll spend the rest of the time here talking about. Now, TYROBP had been observed by in the original paper by Ben, and also by VNA in our group, to be elevated slightly in the brains of patients with late onset Alzheimer's disease. But the real sort of convergence on this pathway came when in early 2013, Ben Zhang reported the DAP12 TYROBP story at about the same time that Alison Goat and John Hardy recognized the TREM2 variants that were linked to Alzheimer's disease and had risk effect sizes approaching that of ApoE. And the reason these two things sort of were an aha moment is that TREM2 and DAP12 TYROBP form a direct protein-protein complex on the surface of microglia. TREM2 provides an ectodomain for sensing the environment, a transmembrane domain, which then associates with the transmembrane domain of DAP12 TYROBP, and that then is connected to the spleen tyrosine kinase SYK. So this is a perception module, the ectodomain of TREM2, signaling across DAP12 TYROBP and to p-SYK. This coordinates then sensing, signaling, and phagocytosis. So the readout of what happens from here is phagocytosis. I think in the interest of time, we'll skip that. So this complex in the microglia sits here at the plasma membrane. The DAP12 TYROBP complex interacts not only with TREM2 but also with CR3, which is also important, as you'll see in just a moment. The readouts are the activity of the SYK kinase, as well as generation of superoxide and activation of the NLRP3 inflammasome. If you just sort of take a larger view of the signaling pathway, again, here is TREM2 and DAP12 or TYROBP, and the plethora of downstream signaling pathways that can be activated or inhibited by this signaling pathway. Some of our recognizable Alzheimer-associated molecules, ApoE, the amyloid beta oligomers, associate with TREM2, and that's how they trigger this signal. The ADAM10 shedding enzyme actually removes the ectodomain of TREM2 as well here, so that also interacts there. There are two basic ways of how this complex is converted into cytoplasmic signals. When there's a multi-site or high valence interaction with multiple TREM2 molecules, that tends to activate this cascade. While when there's only a monovalent stimulation or a low affinity, that tends to inhibit these same various pathways. So our work in the validation phase of AMP-AD, again led by Michelle Ehrlich and Vinay Oray-Mirand, has been to cross these hits as they come out of the computational analysis, like for example TYROBP, to either knock out or overexpress in various systems that seem to express phenotypes relevant to Alzheimer's disease, and that is mice that are either engineered to accumulate amyloid or tauopathy, PSAPP mice, the Borchardt mice, or the P19 tauopathy mouse. So they were either TYROBP was either knocked out or later overexpressed. We also had some effort in flies for a while that since has not been so useful for studying the immune system because the fly immune system is so different. But we do have a collaboration that's been very productive with New York Stem Cell Foundation, with Valentina Fossati and Scott Noggle for studying this in iPSCs as well. Again, looking for the effects of these emerging hits from AMP-AD from the computational analysis and see how they might tweak or not standard phenotypes for Alzheimer's disease. So the first question we asked in these mice, since TYROBP levels are elevated in the Alzheimer's brain, the phenotype is predicted to be at least partly ameliorated in the DAP12 TYROBP knockout. So that's a question. That's a important clinical question. But another question was, does deletion of this molecule change the DAP12 TYROBP network? So the network that I showed, that Ben discovered and reported in Cell in 2013, is a predicted network that's based on static measurements of RNA sequencing and molecules where these hubs and drivers go up and down, and they take 50 molecules up and down with them, and that's the network. That's what network behavior looks like. That's how the network is defined. That's not necessarily a biologically relevant construct. That's just an analysis of static data. But the ability to tweak these hubs and drivers from the networks to tweak them in mice and then redo the multiomics will tell us whether they actually function as network hubs and drivers as they're predicted from the postmortem human material. So the first couple of papers on TYROBP came out in 2017, and then there were two papers back-to-back in molecular psychiatry and on the cover of molecular psychiatry in 2019. And DNA was first author on two of these. And here, TYROBP was deleted from first the TYROBP knockout mouse itself was characterized, and then it was crossed with these plaque formers or tauopathy formers. First of all, despite the fact that I told you how important oligomers are, the TYROBP tweaking did not have any effect on oligomeric A beta levels in these mice. These NU4, A11, and OC are oligomer strains or subtypes. The overall amyloid burden was not modified. The overall microglial density was not modified. What's modified is what you can see here, and that is the plaque association of microglia. And that seems to be attenuated in the absence of TYROBP. You see that a little more obvious here. This is with intact TYROBP. You see the microglia associated with the plaque here, but this microglia in the TYROBP knockout really only has one pathetic little microglia attached to it. Otherwise, it seems to be being ignored. So something about the attraction or the interaction of microglia with with plaques is what seems to be TYROBP dependent. It also plays a role in plaque compaction. This is shown here in sort of wild type TYROBP background versus to the far right, the TYROBP knockout background. The plaque density is much diminished in the absence of TYROBP. This is just more of the same figures just to show the TYROBP knockouts here compared to the TYROBP intacts up here. The microglia studying the plaque in the wild types, but very few microglia in the TYROBP knockouts. So the question then, a question was, when we look at the multi-omics, the computational analysis of these mice, do we see that the network predicted by the postmortem human data is in fact functional? Is TYROBP a driver as predicted, or a hub and driver as predicted by Ben from the postmortem human work? And yes, indeed, it was. And as you would expect, or as you might hope, the polarity was exactly the opposite. All the things that went up in the Alzheimer brain went down when TYROBP, when the hub and driver was deleted. So that then confirmed that this was a biologically active, meaningful network, not just a static sort of reevaluation of genetic changes that weren't related. This was a sort of a living network. Curiously, and this was unexpected, but has turned out to be one of the most interesting parts of the story, is that in the TYROBP knockouts, both the plaque formers and the tangle formers, or the tauopathy that mice that you'll see in a moment, the deficiency of TYROBP allowed the mouse to retain normal learning behavior and normal synaptic physiology, despite the fact that it was accumulating all this Alzheimer pathology, and it apparently was resistant to the accumulation of this Alzheimer pathology. I think you can maybe look here at these electrophysiological tracings. You can see that basically the Borcheldt mouse, the amyloid mouse, the tracing is here, while the wild type and the combination of the Borcheldt plus the TYROBP knockout is here. Basically, they retain wild type electrophysiological function and learning behavior. And so the question is, why is that the case? And the case, that seems to be the case because of the effect of TYROBP deficiency on C1q. C1q seems to be, the levels of C1q seem to be under the control of TYROBP, and they seem to be deficient in these when TYROBP is knocked out. Why do we think that's important? And that's shown here in a paper in Science from Dennis Selko and Beth Stevens. They described this reaction here. And this we think now is an especially important part of the Alzheimer's story that is not necessarily going to be obvious by amyloid scans, for example, or even A-beta levels. This is synaptic phagocytosis by microglia. And this is stimulated by oligomeric A-beta, C3, and C1q. So we think that TYROBP deficiency is sparing the electrophysiology and learning behavior of these plaque-forming mice by removing C1q. Sorry, I can't see where my arrow is. By removing C1q, therefore precluding microglial phagocytosis and sparing the synapses. This reaction was first reported by, part of the involvement of C3 was deduced by Cindy Lemire and Beth Stevens, who looked at C3 knockouts and found that they too, like TYROBP knockouts, their synapses were protected from microglial phagocytosis. ApoE4 seems to also promote this. In the interest of time, let me just say that in the tauopathy mice, the tauopathy actually gets worse, but the function is again preserved, again because C1q is depressed, is reduced. So we think that this is really an important part of the story, the C1q modulation of synaptic phagocytosis by microglia. Let me just spend the last few minutes on a study that was reported in Alzheimer's and dementia at the end of last year, looking at this sort of taking TYROBP into the story of disease-associated microglia. This was a term characterized, at least in degenerative diseases, by Ido Amit in 2017 and 2018. And there are a series of microglial molecules that turn out to be genes related to Alzheimer's disease, but that are induced as microglia interact with amyloid plaques. And this is these are the events that Mikhail Ardrain characterized together with Vianney and Michelle at the end of last year. So these DAMs, disease-associated microglia, are defined by their transcriptional profiles. And these transcriptional profiles are transcripts of genes that are related to Alzheimer's disease. ApoE, TYROBP, TREM2, progranulin all play roles. CD33 play roles. These all increase the risk. ApoE2 decreases the risk. These are all part of the definition of the DAM phenotype. CD33, PCAL, BEN1, these also are all playing a role. So the DAM, several high impact DAM papers emerged over the past few years, focusing primarily on TREM2. And it's clear that there is TREM2 is important, but as you saw from when you saw this diagram before, TREM2 only interacts with signaling across DAP12 TYROBP. So we thought, of course, this must be playing a role in DAM signaling as well. And this is the masthead of Mikhail and Vianney's paper with Michelle from the end of last year. And the highlights are the following. So the first sort of unexpected event was that a key part of the DAM phenotype is transcription of ApoE. There's been lots of argument over which cell types in the brain express ApoE. Everyone knows astrocytes do, and they were responsible for the bucket of expression, the bulk of expression. But it's very clear that microglia do as well, and that the absence of TYROBP, the induction of ApoE in pori-plaque microglia is very much impaired. And so this shows the induction. So these the red microglia here are expressing ApoE. All right, and this is the wild type plaque-forming mouse. If you now delete TYROBP, those cells are gone. Or that trend, now the cells are there, but the transcript is gone, the red signal is gone. The ApoE mRNA is no longer induced. So this is part of the DAM phenotype, and it's very clear that TYROBP is also contributing to the DAM phenotype. Absence of TREM2 did not prevent the increased peri-plaque ApoE in these mice the way we saw with TYROBP. This is really the same sort of figure that you saw before, but now these are CRND8s that are deficient in TREM2. These were created by Paul Fraser and Peter Hislop, and the ApoE, the peri-plaque ApoE is still persistent in the absence of TREM2. Michelle and Mikhail created mice that overexpressed TYROBP specifically in microglia, and studied those mice as well. And here the phenotype is as you would predict. They now show decreased plaque density and decreased microglial reactivity. when we overexpress TYROBP in the tauopathy mice, in the amyloid, in the absence of pathology, this now again looks like it seems to make the tauopathy worse, and this has led us to this sort of conclusion that among a number of genes with a sort of inverted U or a Goldilocks effect, where genes seem to be expressed at optimal levels, and either too much or too little can increase disease risk. Finally, just to sort of wrap up the issue of whether TYROBP, the evidence is, the suggestion is that maybe it's a resilience gene, but is this something that only happens in age? And so at 8 months of age in TYROBP, the DAM phenotype, the DAM genes are unchanged. But by 20 months, as aging begins or as aging happens, the expression of DAM genes is down-regulated. So this now is consistent with the notion that that TYROBP may be a resilience gene, both at the level of synaptic phagocytosis and at the level of converting microglia into the DAM phenotype. We're now in the process of, Michelle and DNA are in the process of creating mice that have conditional knockouts in TYROBP, so as to be more sort of drug-like, to see whether that would be something that we can then pursue therapeutically. So I'm going to stop there. There are several stories that are very exciting in the lab I haven't gotten to get to. One of these that's emerging now, some papers have emerged, others that are currently being developed with miR-155 and the INPP5D SHIP1 network. That is the ability of this microRNA and this inositide phosphatase to modulate the inflammatory activity of microglia. So I think what I want to do, I'm going to skip the neurogenesis story. And I just want to close by thanking everyone who's actually done the work here. Sorry. So in addition to those stories I didn't get to touch, VGF emerged as an important AMP-AD target. Steve Sultan has devoted his career to characterizing VGF, and he, Michelle, Eric Schatt, Ben Zhang worked on both this omics characterization, the discovery of the network, the VGF network, and its study in mice. So again, as I've said throughout, Michelle, together, especially with VNA, have led much of this work in microglia over the last few years. The AMP-AD program, pioneered by Eric Schatt, now led by Ben Zhang, a major contributor to the computational analysis of Mingli Wang. Ben Reedhead, whom I haven't acknowledged enough, has been very much involved in the inflammatory interactions, especially with the viral networks. Masude Bichak has taken over with Ben, or in collaboration with Ben, in studying this neurogenesis story that I wasn't able to squeeze into this hour. But I think we're almost at the hour now, so maybe I will stop and ask for questions.
Thank you, Sam. Maybe you could unshare the screen. Yeah, we're at the top of the hour, so we'd have time just for a question or two, if anyone's got a question. If you do, please feel free to unmute.
Can I ask a quick question?
Yes, indeed, Rob.
Yeah, hi Sam, great talk. Quick question, the tau toxicity in humans, it's thought to be downstream of the amyloid plaques. Now you have the Dutch model and the TYROBP mutants, very interesting changes in the amyloid. Can you in your mouse models look at tau, phospho-tau, or are mouse models not suited for that?
No, it's possible. It's just that it's, we're trying to isolate specifically the mechanism of how oligomers are toxic. So we certainly agree that oligomers and fibrils can propagate tauopathy and that contribute to the human disease. The issue is where very early on, there's a specific toxicity attributable to the oligomers that's just not part of this inflammatory tauopathy story. That's a separate issue. And we're just trying to isolate that very earliest step to try and see if we can intervene there and prevent all the other downstream stuff from happening.
Thanks.
Can I ask a question?
Yes, go ahead, Anna.
Hi, Sam. That was a great talk. Thank you so much. Quick question. Can you comment the difference between the monoclonal antibodies against amyloid in terms of affecting the oligomeric species, which is the most toxic?
Yeah, so Mary and I were talking about this earlier. We're really hamstrung by the inability to detect and even distinguish toxic oligomers versus neutral oligomers in the living brain. So we know these are claimed to be anti-oligomer antibodies because that's how they were developed. They were screened. The immunogens were oligomers. The Arctic A beta makes oligomers. The P3 truncated pyroglutamylated A beta forms potent oligomers. So they were oligomeric at their design, but we don't have any way of seeing even the whole range of oligomers in the living brain. We can't see oligomers at all. So the part of the problem of getting a sense of toxicity is that we still can't see the most important poison. And we also know that there's a range, just like with prions, there's a range of oligomer strains. Some are not so bad, probably, and some are maybe even are clearly more potent than fibrils. So unfortunately, getting those data from the living brain is still a challenge. You're stuck by saying that saying what your reagent was characterized to do and what it does biochemically, and imputing what it does in the living brain, but not being able to connect all those pieces. Now, if this microglial synaptophagy and synaptic density is key, maybe this new PET ligand that visualizes synapses will be a useful surrogate for, a better surrogate for what's going to happen clinically than amyloid.
But out of the monoclonal antibodies that have been tested, the SOLA, the bapineuzumab, donanemab that is on the trial, and now the aducanumab, they target the different species of amyloid, right? Do you know if there's a ratio of preferential oligomeric targets?
So the problem with those in particular is that they're all proprietary, and so this now means someone getting their hands on the actual material from each one and comparing against some standard panel of oligomers. So that's potentially knowable but unknown at this point.
Thank you.
Okay, it's 4 minutes after the hour, so I think we should end there. Thank you so much, Sam. That was great. Thanks everyone for joining us, and we'll see you at the next seminar. Thanks very much.